Just a reminder, if you haven't turned it in already, your chapters 3 and 4 outline, I just kind of mushed them together. So if they're a little bit shorter than normal, that's fine. Those are due today. So turn them in if you have not already. You can just turn in one document for that. And a reminder, your first exam is Friday. So everyone will be in person Friday. So for you guys on Zoom, you guys are on Zoom for Monday and Wednesday, but then Friday, you'll be in the labs, the bio labs upstairs to take the in-person exam. So everyone will be in person. I'll talk more about the layout of the exam, and we'll do a quick review on Wednesday. But we'll be able to spread out across 2 lab rooms so we can have social distancing and still do the exam in person. There's just not enough room in this classroom. Okay, any questions about anything before I get started? Nothing. Okay. So let me share my screen here. So we're going to finish up. So on Friday, we did that cell organelle activity. So the information that you need to know, the slides basically, are at the end of this chapter's presentation, the PowerPoint file. So the chapter 3 cell structure lecture is where you'll find at the very end the details that you need to know about the organelles. Hopefully that activity, I know you guys were on Zoom, you guys in the classroom, hopefully it was still kind of helpful as a method of sort of attaching some kind of meaning to these little tiny organelles that are within our cells. I think we ended here or right before here last time. So we were talking about the phospholipids and all the lipids in the cell membrane, right? So we talked a little bit about the cell membrane with the organelle activity. I'm going to go into more detail today because there's a lot of info you need to know about the cell membrane just because it's what keeps the cell in balance. So as far as what's coming in and what's going out, there's all these proteins essentially that are allowing certain things in and keeping other things out and vice versa. So proteins are really important in the cell membrane. They only make up 2% of the molecules, like by number, by count, but they make up about half of the mass. So they're big and heavy, but there's not that many of them. But they play a very important role in letting material in and out of the cell. So that's what I'll talk about for the first part of class today. We'll spend most of the time on that. And then I'll start to get into chapter 4, which is also on your exam. And that deals with DNA, RNA, translation, transcription, how we move from DNA to proteins, basically. And however far we get through that on Wednesday, that's what's going to be on your exam. I don't know that we'll get through all of the info. The end of it is mitosis, which you'll be doing in lab. So you'll at least get that information in lab. So we'll see how far we get. And I will leave time on Wednesday to do a review, no matter what. So we'll see how far we get. But I want to spend most of today talking about the cell membrane, as exciting as that sounds. Okay. All right, we're talking here about the transport across the membrane. That's really why we care about the cell membrane, because we care about what goes across it and how it goes across it. You can think of the plasma membrane or the cell membrane as selectively permeable, which just means there's certain things that will go through it and certain things that won't. A lot of it depends on the characteristics of the molecule, as well as how much is already in the cell versus how much is outside of the cell. So the relative concentrations play a big role as to what will pass through. I'm going to talk about sort of 3 different categories of membrane transport today. The passive mechanisms don't require energy, so no ATP is required for passive. This includes filtration, diffusion, osmosis. Diffusion and osmosis hopefully sound kind of familiar. Filtration, I'll touch briefly on the next slide. Active mechanisms are anything that consumes ATP. There's a couple different kinds of those that I'll talk about. So that's going to use energy. And then you have this sort of separate category that can be either active or passive, and that's carrier-mediated transport. So that's really anytime a protein is used. So those proteins I showed you on the previous slide, anytime a protein is used to move something from one side of the cell to the other, one side of the membrane to the other, that's going to be carrier-mediated transport. It can be active or it can be passive. So sometimes those proteins need energy and sometimes they don't. So I'll talk about each of these main bullet points. And that's basically, that's the aspects of the membrane that I want you to understand, how stuff moves across it and why and when it will move across it. So first, filtration. You may not have heard of filtration before. This is just where pressure is actually pushing material through cell membranes, or in this case, actually through little breaks between cells in our blood vessels. So this is all due to pressure. So you know your blood has pressure running through your veins, right? Kind of like a hose. And that pressure is basically pushing little solutes out of the capillaries. So these little colored balls are being pushed out, water, solute, that kind of stuff is being pushed out of the blood capillaries. That's all filtration is, and that's really all I want you to know about filtration. It's being pushed out by pressure. It's all driven by physical pressure. It doesn't matter how much solute is in the blood capillary versus how much is outside of it. That'll come into play in a large way in just a minute. Okay, so filtration, pretty simple. Simple diffusion is also pretty simple. I think most people have heard of diffusion. There's simple and there's facilitated. So simple diffusion is just where you have molecules moving from one side of a membrane. So this little pink line here in the middle is going to be our selectively permeable membrane. Molecules are moving from where there's a lot of them to where there's fewer of them. So high concentration to low. That is down the concentration gradient. And that just happens naturally due to molecules moving around and bouncing off of each other. They're just going to move to where there's fewer of them and spread out, so they're a little bit more equalized. That is simple diffusion. There's no protein that's needed. There's no energy needed. It's simple. So just remember, down the concentration gradient from high to low. That's simple diffusion. And osmosis is specifically dealing with water for our purposes. So this is the movement of water across a selectively permeable membrane. So water is going to move from where there is a lot of it to where there is less, just like simple diffusion. So you can think of water, we don't often think of the individual molecules of water, but think of individual molecules of water. If there's a lot of them on one side and not as many on the other side of a membrane, the water is going to move to where there's less water. Maybe a more applicable way to think of this is that water is going to move towards where there's more solute. So if there's a lot of salt on one side of a membrane, water is going to move that way. It wants to dissolve the salt. That's kind of how I always think of it. Let me pull you Zoom people over here. Okay. So if we're looking at the left side of this beaker, these little blue circles are going to be water. The red circles on the other side are solute, some kind of salt, we'll say. So water is generally going to move to the right. So So for you guys on Zoom, if you couldn't see that, water is going to move from side A to side B because there's more solute on side B. And over time, the water level on side A will drop because a lot of it's moving over towards the solute. So that's the easiest way to think about it, I think, especially because I'm about to get into how water moves in and out of cells and what that does to the cell. So it's going to follow, it's going to go towards where there's a higher solute concentration. This is really important for IV fluids, as I've noted here. So if someone gets an IV and it doesn't have the right amount of salt to water, it can really cause some major balance issues, osmotic balance issues, and cause some major harm. So IV fluids have a very specific amount of salt in them. So it won't cause a massive movement of water into or out of our cells once that IV is put into our body. Osmotic imbalances also underlie a lot of common conditions: diarrhea, constipation, swelling, or edema. So if you have swelling in part of your body, that may mean there's too much solute concentration there and water is kind of pouring towards it. So osmotic balance is really important sort of throughout our body. Okay, so remember, osmosis, movement of water. The solute isn't moving when we're talking about osmosis. It's really just the water that's moving. And on the next few slides, I'm going to talk about the movement of water specifically. So we're going to stay on that train for just a little bit. Hopefully these words look kind of familiar. Hopefully you've seen them before. Hypotonic, hypertonic, isotonic. These terms refer to a solution. And for this, I think the easiest thing to imagine is a cell, we'll imagine a red blood cell, sitting in some kind of watery solution, like a bath. So these terms refer to the solution that that cell is sitting in. So does it have more solute, less solute, or the same amount of solute as what's inside the cell? That's what we're talking about here. What are the relative concentrations of solute inside the cell versus in the solution that the cell is resting in? Depending on what that answer is, the relative concentrations, water is either going to move into the cell or it's going to move out of the cell. So we'll start with hypotonic solution. Hypo means low. So if you're hypoglycemic, your blood sugar is too low and you may pass out. Hypo always means low. So this solution has a lower concentration of solute in the solution around the cell than the cell has. So the cell has a greater solute concentration. That's what I've noted here. So more solute, more salt inside the cell compared to outside. So try to picture that in your head. I'm really visual, so I always try to picture things. So lots of little dots of solute inside the cell, not a whole lot outside. That's basically going to mean the water is going to flow into the cell, right? Because there's a lot of solute in the cell, so that water is going to move towards that higher concentration of solute. And I'll show you an image of this in a couple slides to compare these three solution types. That will cause the cell to swell because you have a lot of water pouring into it. So the cell will kind of grow and swell and may burst if there's too much water coming in. All right, so that's number one, hypotonic. You can probably guess that hypertonic is going to be the opposite of that. So hyper means high. Hyperactivity means somebody has too much energy, right? So hypertonic solution means that the solution around the cell has a high amount of solute, a high solute concentration compared to what's inside the cell. So now that means water is going to leave the cell, right? Because there's a lot of solute surrounding the cell. The water is going to want to move towards that. So it'll be sucked out of the cell, and it can cause the cell to shrivel. So now our solute concentration inside the cell is less than the solute concentration outside the cell. And then isotonic is really easy. Iso means the same. So the solute concentration inside and outside the cell is the same. Water moves in and out, but it equalizes on both sides. So there's no change in cell volume. It doesn't expand, it doesn't shrink, it just stays the same. And going back to the idea of intravenous fluids, IV fluids, that's what the goal is with IV fluids, is to basically create an isotonic solution. So something that matches what your body fluids, the solute concentration of your body fluids. And that's actually 0.9% sodium chloride. So most of your body fluids are about 0.9% sodium chloride. So that's what normal saline is. That's what's coming out of that bag. If you've ever seen an IV bag, it's not pure water. If it were pure water, it would cause the cell to shrivel down, or sorry, expand and potentially explode. So then the concentration of the IV fluid going into your body and the fluid already in your body is pretty equal. So there's no real expansion or shrinking of cells. Okay. If it's not 0.9% NaCl, if it's too high or too low, it can cause some pretty major issues, as you might imagine. All right, so those are the three types of solutions that a cell can be in. I think these images are kind of helpful. Like I said, I think pictures always help me personally. So if we think of, we'll do hypotonic first. So our hypotonic solution, this brown thing is our cell. That's the membrane. Make sure you guys can see. These little green dots are the solute particles. So you can imagine NaCl if you want. So because there is more solute in here, there's water on both sides, but water is going to go towards that higher solute concentration. And you can see here on this red blood cell example, water is going to pour into the cell. It'll make it expand and potentially explode, which is not good. An isotonic solution, easy, water is going in and out at the same rate. You don't get any change in cell volume. And then hypertonic, you have a lot of solute outside the cell and not as much inside, which means water is going to move out, right? So water is moving out of the cell, causing that cell to kind of shrivel down. So this is what the different kinds of solutions will do to a cell, hypo versus hypertonic and isotonic. And if we look at some scanning electron micrographs of a red blood cell, rather than just a drawing, you can see that those same examples with the microscope. So you have a hypotonic solution where a lot of water is flooding into the cell and expanding it. Isotonic, red blood cells have this little like indentation naturally in the middle. So if it's isotonic, you can still see that indentation. It's its normal shape. Hypertonic, you have this shriveled up shape because water is flowing out of the cell. So for you guys on Zoom, if you couldn't see that, this is the expanded hypotonic solution. The isotonic solution, you have that red blood cell with the little normal indentation. And then hypertonic, you have this shriveled up kind of blood cell because all the water, not all, a lot of the water has flowed out of it. Okay. So you can see why the type of solution that a cell is in matters, right? We don't want all of our cells shriveling up or expanding and bursting. Any questions about those three terms? Keep them separate in your mind and be able to tell me if water is going to move into or out of a cell based on the type of solution it's in, and what the consequence may be. So if I give you a solute concentration outside the cell versus inside the cell, let me know. You'll have to tell me if water is moving in or out and what that might mean for the cell. Is it going to expand? Is it going to shrivel? Is it going to stay the same? Okay. Now, going back to the proteins. So that was all, a lot of water will move across cell membranes without the help of proteins. So we weren't really focusing on the help using those proteins yet. So now for this last section of this chapter, I want to focus on how cells use proteins in their membranes to get material in and out. So that's carrier-mediated transport. Transport proteins are carrying solutes in and out of a cell. And we can break down carrier-mediated transport into 3 different categories, one of which is passive, 2 of which are active. So passive carrier-mediated transport, so a protein's involved, but no energy is needed. So the protein sort of just acts like a channel almost to allow a molecule to get into or out of a cell. This is necessary for a lot of molecules just because it's not always easy for a molecule to move through the phospholipid bilayer on its own for multiple reasons. It might be too big. It might not be able to mix with this hydrophobic internal section of the phospholipid bilayer, et cetera. So sometimes you just need a little channel to let molecules in. That's going to be facilitated diffusion. So the diffusion we were talking about before was simple. No protein was needed. Now facilitated diffusion, that protein is facilitating or helping a molecule move across the membrane. And then we have the primary and secondary active transports. So with facilitated diffusion, since it's passive, it's still moving down the concentration gradient, that molecule it's moving from high to low concentration. The 2 types of active transport obviously need some kind of energy because the molecules are moving against or up their concentration gradient. So they're moving from areas where there's not a lot of them to where there is a lot of them. So they're moving in the opposite direction that would naturally happen. So you need energy to make that happen. So here you can see energy being used on this protein to pump this little molecule out of the cell against its concentration gradient. So here we have energy being used to pump that molecule out of the cell. Primary and secondary active transport are really similar. This is going to use energy directly. This will use it indirectly. So primary means the energy is being used directly, and I'll show you what that means in a minute. Secondary active transport, the ATP is being used indirectly, and that's kind of hard to wrap your head around, but I'll show you what that looks like in a minute too. All right, so let's get into, that's all I'm going to say about facilitated diffusion, by the way. I'm not going to go into detail on facilitated diffusion. We're going to focus on the 2 types of active transport here. Okay, so primary active transport, we know that a carrier is being used and the molecule that's being moved up its concentration gradient. So the protein that is moving that molecule, that membrane protein, is directly using ATP to make that happen. So that protein is basically breaking down ATP to use it to move the molecule into or out of the cell. You may have heard of the sodium-potassium pump if you've taken a general bio class. Hopefully you've heard of it. It's a really important pump in pretty much all of our cells in our body. And it's going to move sodium and potassium in opposite directions, but both against their concentration gradient. So the goal of these active transport mechanisms, especially primary active transport, is to build up sort of an unnatural concentration of some kind of solute. So I'll show you the sodium potassium pump, but the goal is to build up a lot of sodium outside the cell. Whereas normally it would just flow back in down its concentration gradient, but the cell is trying to basically push a lot of sodium out of the cell. The calcium pump, similar idea. We'll talk about the calcium pump when we get into the muscle unit. So calcium's being basically moved to an area where it's already highly concentrated. So that's, again, against its concentration gradient, that requires energy. So now I just want to look briefly at the sodium potassium pump and kind of the overall idea of it and how it then relates to that secondary active transport. All right, so sodium potassium pump, primary active transport. I took some of the details out of this just so you guys sort of have a general overview of what's happening, like the important parts of it. The idea is to keep a lot of potassium, these are ions, so K+, potassium ions in the cell, and a lot of sodium ions, Na+, outside the cell. So we have building up the concentration of sodium outside the cell, building up the concentration of potassium inside the cell. So the pump will basically move each in opposite directions, kicking sodium out and bringing potassium in. And ATP is used for every round of that. An interesting factoid here is that this pump actually uses about half of your daily calories that are burned. So you burn X amount of calories during the day. The sodium potassium pump in all of your cells is using half of those. So something we don't think about. But it takes a lot of energy to actually create these concentration gradients. So there's a couple of goals. So why are our cells always pumping sodium out and potassium in? What is the point of that? These are the two main reasons for why we want more sodium outside our cells and more potassium inside. So for one, it maintains this really steep sodium gradient outside the cell. That's going to allow for secondary active transport to happen. And I'll talk about the secondary active transport in just a minute. But having a lot of sodium outside the cell will allow for other active transport mechanisms to occur. Something we won't get into for a little while, it actually maintains a negative charge inside your cell. So inside your cell is negatively charged compared to all of your cells, compared to outside. And that sort of sets up a situation where you have all this potential energy. You can think of that negative charge inside your cells as potential energy. And that's what's going to allow your muscles and nerves to fire. If you didn't have that negative charge inside your cell, your nerves wouldn't be able to fire and your muscles wouldn't be able to fire, because it's all just electricity basically that's causing our muscles and our nerves to fire. That negative charge is that form of electricity that can be used. So it maintains that negative internal potential within the cell because, as you notice, so these are both cations, they're both positively charged, but 3 sodium ions are going out for every 2 potassium ions coming in. So because you have 3 positive charges going out and only 2 coming in, each time that pump moves 3 sodiums out and 2 potassiums in, that brings the charge down just a little bit inside the cell each time, right? Because you're getting rid of more positives than you're bringing in, which lowers the charge in the cell. So those are the 2 primary reasons why the sodium potassium pump, you'll hear about it in a lot of different biology classes. That's why it's so important. It allows so many other things to actually happen in our bodies. Okay. Now, so what is secondary active transport? If primary active transport allows for secondary, what is secondary? This is where a carrier is going to, again, move solute through a membrane, but use ATP indirectly. So the solutes that are moving through and the protein that's doing it, that protein isn't directly using the ATP, but the process needs a primary active transporter to make this happen. So what do I mean by that? The example I'm going to give you is the sodium glucose transporter. So you can see over here, we have this SGLT protein. So this is our transporter. These green hexagons, 1, 2, 3, 4, 5, 6, yeah, hexagons, are glucose. And then these little reddish orange dots are going to be our sodium. So glucose and sodium. Down here, we have our sodium potassium pump. So up here, we have our sodium glucose transport for you guys on Zoom. Down here, our sodium potassium pump. So they're on opposite ends of the cell, right? The sodium glucose transporter, you'll see, does not use ATP directly. There's no ATP coming off of it like there is down here with the sodium potassium pump. But you can think of sodium as sort of a way to bring glucose back into the cell. So glucose is sort of hitching a ride on sodium and flowing back into the cell. And the reason sodium is coming back into the cell is because there's a lot of it outside the cell and it naturally wants to flow down its concentration gradient. So it's coming back in. Without the sodium potassium pump increasing the concentration of sodium outside the cell, you wouldn't have a high concentration to allow for that. Does that make sense? So there's a lot of sodium outside the cell only because of the sodium potassium pump, which uses energy. And then glucose, like I said, hitches a ride sort of on the sodium. So the glucose is hitching a ride on the sodium to be brought back into the cell. But no energy is directly used for that protein. So in our kidney cells, these are pretty prevalent, the SGLTs, along with the sodium potassium pumps on the other side, because otherwise we lose a lot of glucose through excretion in our urine, and we need glucose. So I should step back. Glucose is critical to respiration of our cells. So glucose is kind of a fuel source for all of our cells. So bringing glucose back into the cell is really important. We don't want to just excrete it all with our urine. We want to hang on to it. So in kidney cells, these SGLTs paired up with the sodium potassium pump are really prevalent. It helps to bring glucose back into the cells so it's not excreted. Okay, any questions about the secondary active transport? Pretty exciting stuff, I know. Okay. All right, and that is all of the membrane stuff. So like I said, I cut some information out of that chapter just to focus on, I think, the really important aspects, because we'll talk a lot about stuff moving across membranes as we get into the muscle and nerve stuff, as well as, nervous system. Yeah. So there's a lot of hidden slides on here. If you download this, you can ignore all of them. And then, like I said, at the end of this lecture is where all the organelle slides are. So you can find all the information on the organelles here, like we went over on Friday. But that wraps up chapter 3 for us, the basics of cell structure. And now we're going to move into chapter 4. Okay. Now, like I said, I may not get through all of this material. Whatever we get through is what will be on the test. If we don't get to something like mitosis at the end, I won't ask you about it on the lecture exam, but we're going to go through mitosis in lab this week, so you will have that info. All right, so there's a lot to chapter 4. Once again, I'm going to focus in on what I think are the most important aspects of chapter 4. So we're going to talk about basically how we move from genes and DNA to RNA to proteins. So that whole process, starting with hopefully kind of a review here on nucleic acids. So these were one of the 4 major biological macromolecules, right? And the nucleic acids for our purposes are DNA and RNA, deoxyribonucleic acid and ribonucleic acid. And we need to understand these nucleic acids, particularly DNA, in order to study genetic disorders, which we're not going to go into in here. That's like a whole nother class, medical genetics. So we won't really delve into many of the genetic disorders. Some of your diseases that you're talking about are genetically based. So you might dip into it a little bit. So you'll learn about that in your project or from watching other people's videos. Some examples are color blindness, which is very prevalent. Cystic fibrosis, which is thankfully not that prevalent. Diabetes mellitus. So type 1 in particular, diabetes, is genetically based. And then hemophilia. So those are just a few. Hemophilia is when your blood doesn't clot. So if you get a cut, you just keep bleeding. So those are just a few examples of genetic disorders you can trace back to the DNA, essentially. And then we have traced back to the DNA and know where that issue occurs. So yeah, getting back into the nucleic acid structure. Nucleic acids are made up of many nucleotides strung together. And I have a picture of a nucleotide, not a picture, a diagram of a nucleotide on the right over here. It's made up of 1 or more phosphate groups, a sugar, a monosaccharide, and a nitrogenous base. We're going to focus on the nitrogenous base as far as DNA and RNA, because we're going to talk about the genetic code. And the genetic code comes from the nitrogenous base that's attached to the nucleotide. Hopefully the central dogma of biology looks kind of familiar. Hopefully you've heard it before, but if you haven't, the whole idea of this chapter really, and we call it the central dogma of biology because this is how the process, a really important process, happens. DNA is going to code for RNA. RNA will code for proteins. So the ultimate goal is to get from DNA to proteins. RNA is kind of the intermediary that has to occur in between that. Okay. So that's kind of a refresher on nucleic acids. So what do I mean when I talk about the genetic code? I'll show you a table for the genetic code and how you can determine what amino acid is being coded for with these nitrogenous bases. But like I said, the nucleotide, the really important part of the nucleotides is the nitrogenous base. There are 5 nitrogenous bases. 2 of them are double ring structures, so lots of carbon and nitrogen in each of these, in all of these. So the purines they have 2 rings connected to each other. The pyrimidines are just 1 ring, single ring. The 2 purines are adenine and guanine, A and G. The pyrimidines are in DNA, cytosine and thymine. And if we're talking about RNA, uracil replaces thymine. So in RNA, it would be cytosine and uracil. It's kind of an annoying switch, but you have to realize whether you're talking about RNA or DNA and if it's thymine or uracil. So what I want you to know from this is which are purines. Know the purines are double rings. The pyrimidines are single rings. Know the names of the pyrimidines. And then I'll talk about the bonding pattern between these. So a purine is always going to bond with a pyrimidine and vice versa. And that base pairing is essentially the genetic code. Okay. So I think most people know that DNA is this double helix structure, right? So it's 2 strands that are wound together into a double helix. Those 2 individual strands, which you see separated over here, are held together by the nitrogenous bases bonding with each other on each strand. So for you guys on Zoom, here we have one half of the DNA molecule, one strand, and the other half on the other side. And then in the middle here, this A and T, those are the nitrogenous bases. So adenine and thymine bond with each other all the time. And then guanine and cytosine. So A and T always go together, G and C always go together. Unless we're talking about when we get into moving from DNA to RNA, the T will be replaced by a U. So those are the base pairing rules, A, T, C, G. And a quick kind of step back looking at what chromosomes actually are. So you've probably heard the term, well, I'm sure you've heard the term chromosome. A chromosome is essentially just condensed DNA. Normally, if a cell is not replicating itself, it looks like this, just kind of looks like a bowl of spaghetti, I guess. So this will be the nucleus of a cell when it's not replicating itself. And it's in what we call chromatin. So it's all stretched out. When the cell goes to copy itself, it will condense into this nice compact chromosome, this X-shaped chromosome. So here you can see a slide of it. So chromatin, you can't really distinguish anything. It just looks like a big blob in the nucleus. Once it condenses into the chromosomes, you can kind of see individual strands of chromosomes. So you know this cell is basically about ready to replicate itself. This one is not. We'll talk about that in lab. One other important term on here is sister chromatids. So this will come back up. Each side of this chromosome, each of these is called a sister chromatid. So one side of the X, each side of the X is a sister chromatid. It has the same genetic information on each side, it's just a copy. So when a cell replicates and copies itself, half of the information can go to one cell and the other half can go to the other cell, so it's an exact copy of itself. And now to RNA. So RNA is just ribonucleic acid. There's 3 different kinds that you'll need to be familiar with. It is single-stranded, so it's not that double helix. It's just one strand. You still have nitrogenous bases on the nucleotides, but remember uracil replaces thymine. That's the only difference. You still have the A and the G and C. And then in order to make a protein, we basically need 3 RNAs. mRNA gets most of the attention. That's messenger RNA. That's what basically gets made from the DNA. And we'll talk about transcription. That's what gets made as a result of transcription. It's taking the message from DNA to the ribosome to make a protein. So it's the messenger. Transfer RNA will bring in amino acids. And remember, a protein is just a bunch of amino acids strung together and then folded on itself, but basically a string of amino acids. So it's bringing in the amino acids. Ribosomal RNA, all you need to know is that's what makes up a ribosome. We won't really go into more detail on that. Okay, so that's RNA. Any questions so far? I know I'm going through this kind of quick. I'm hoping a lot of it's kind of a review. If it's not, come talk to me at some point, or definitely delve into this chapter in a little bit more detail if you're like, I don't know what we're talking about. All right, and real quick, some information about genes. Everyone's heard the term gene. What exactly is a gene from a physical perspective? It's just a segment of a chromosome, a segment of DNA. So here, if we take this chunk out, that might be one gene here, one gene here. So it's a sequence of nucleotides. That's all a gene is. And then it will code for 1 or more proteins. As humans, we have about 20,000 genes. And if you look at all of our genes at once, that's called our genome. And I'm not going to get into more detail on genome specifically. but just all of our genes are considered our genome. And our genome is made up of, we have 46 chromosomes. So in every single cell in your body, for the most part, you have 46 chromosomes. 23 are from your mom and 23 are from your dad. So every skin cell, every liver cell, every cell that makes up your eyeball, all your bone cells, you have 23 copies of DNA from your mom and 23 from your dad. And then one more slide, and this will wrap us up. So this is the code that our bodies use to produce the proteins that make us, essentially. So you see the nitrogenous bases, the U, the C, the A, the G. So we're talking about RNA here. That's why we have a U instead of a T. This code is read in triplet. So 3 letters or 3 nucleotides will code for 1 amino acid. And we'll look at that in transcription or translation on Wednesday. So you have to have a combination of 3 nucleotides to code for a specific amino acid. There's only 20 amino acids. So there's some repeat among the code. And yeah, so these are the amino acids. Sorry, I lost my train of thought there. So this would be, you don't have to know the amino acids, but it's just an abbreviation: leucine, proline, serine. So all of the amino acids have a very specific set of 3 letters that are necessary in that sequence in order to bring that amino acid and attach it to the growing amino acid chain that will be the protein. And interestingly, pretty much all of life has the same genetic code. So an elephant, a dog, a shark, all of them use the same code. So CUU will code for leucine in pretty much every living organism, including us, which is kind of cool. All right, and that is it. We'll get into transcription and translation on Wednesday and then do a quick review for the exam. So I'll see you then, or I will see you in lab before then, potentially.